step-by-step guide to primary cell culture. Creative Biomart. Principle and Protocol. Introduction. To master the basic operation process of primary culture and subculture of mammalian cells, lay a foundation for the application of bioengineering in medicine. Creative Biomart. The main reagent. 1. 5% calf serum in medium. 2. 0.01 mol/LPBS. 3. 0.25% trypsin-0.02% at a mixed digestion solution. 4. 75% ethanol. Creative Biomart. Major equipment. 1. Surgical instruments. 2. Plates. 3. Bottles. 4. Sucker. 5. Centrifuge tubes after sterilization. 6. Alcohol lamp. 7. Beaker. 8. Clean bench. 9. Carbon dioxide incubator. 10. Inverted microscope. Creative Biomart. Experimental materials. Rabbit, HeLa cells, human cervical cancer cells. Creative Biomart. 1. Primary cell culture. 1. The rabbit was quickly killed by cervical dislocation. Then, the entire animal was immersed in a beaker containing 75% ethanol for several seconds. After removal, it was placed in a large petri dish and carried into a clean bench. The sterilized skin was excised with iodine and ethanol using sterile scissors, and the liver or kidney was removed by laparotomy. Place in a sterile petri dish. Creative Biomart. 1. Primary cell culture. 2. The excised organs were washed 3 times with sterile PBS, and the tissue was carefully cut with an ophthalmic surgical scissors until it became small pieces of about 1 mm. 3. Washed with PBS until the tissue became white. Move into a sterile centrifuge tube and let it stand for a few minutes to allow the tissue mass to naturally settle to the bottom of the tube and discard the supernatant. Creative Biomart. 1. Primary cell culture. 3. Pipette 1 ml of 0.25% trypsin-0.02% at a mixed digestion solution into the centrifuge tube, mix with the tissue block, add the tube plug, and digest it in a 37 degrees Celsius water bath for 8 to 10 minutes. Shake the tube every few minutes. Bring the tissue into full contact with the digestive juice, stop and suck the supernatant. Add 5 to 10 milliliters of medium containing 5% calf serum to the centrifuge tube, mix by pipetting, and move into 2 culture flasks and incubate in a CO2 incubator. Creative Biomart. 2. Passage cell culture. 1. Take out the monolayer of primary cultured cells or HeLa cells from the CO2 incubator. Pour out the culture solution from the flask in a clean bench and add a little digestive juice. It is better to cover the cells with liquid surface and let it stand for 5 to 10 minutes. Creative Biomart. 2. Passage cell culture. 2. Observe the digested cells under an inverted microscope. If the cells become gardens and they are no longer connected to each other, then the digestive juice should be immediately discarded at the super clean bench, and 3 to 5 milliliters of fresh culture fluid should be added and blown to make cell suspension. Creative Biomart. 2. Passage cell culture. 3. Aspirate the cell suspension by about 2 milliliters and add it to another flask. Add 3 milliliters or so of the culture solution to each flask, cap the stopper, return it to the CO2 incubator, and continue the culture. Creative Biomart. Contact information. Email, contact@creative-biomart.com. Website, www.creativebiomart.net. Address, 45-1 Ramsey Road, Shirley, New York 11967, USA. Contact us.